Next steps, we will talk about the process of causal inference. In this figure, in the below here, we can see the process from association to causation, which will exclude the bias and the confoundings, as well as the chance findings. So actually, there are 4 steps for this causal inference process. First, we'll find that the association is actually present and is statistically meaningful by a statistical test that rules out the inference of the chance. Second, the association is not spurious, that is a false association. The spurious association, as a so-called non-causal association, is attributed to the selection bias, information bias, or measurement bias, and the confounding bias. The confirmatory criteria for the causality are satisfied, and then this is the third step. Even if a statistical association does not and it's not due to the bias, causal inference cannot be made confidently without satisfying this confirmation of the criteria of causality. And these causality criteria will be shown in the next slides. The final part is to see whether it's an indirect association or direct association. The term indirect association may be sensed in a broader sense. Indirect causal association is inferred when the risk factor or independent variable causes change in dependent variable or conditions through the mediation of other intermediate variable or conditions. Now let's see the criteria for the causal inference. The first we have to mention is about the Cox prostitute that was assume a particular disease has one cause and a particular cause results in one disease. He stated that the 4 postulates should be met before the causative relationship can be accepted. First, the organism must be consistently present in the disease tissue. Second, the organism must be able to be isolated and grow in a pure culture. Third, the pure culture must be shown to induce disease experimentally. And fourth, the organism must be recoverable from the experimental infected host. As many of you may consider that Cox postulate was very ancient word and it will not affect the current situation. However, actually, the virus that causes COVID-19 was discovered through different scientific evidence that also shown that the virus fulfills Cox postulate and demonstrating that it is indeed this virus was the cause of the COVID-19. There are several causal criteria that are shown in this table and stated by various persons or agents, such as a commonly used set of criteria was based on a list of consideration proposed by Austin Bromfield here. Those criteria expanded upon a list of offered previously in the landmark U.S. Surgeon General's first report about smoking and health. He emphasized that causal inference cannot be based on a set of rules. Condemned emphasis on statistical insignificance test and recognized the importance of many other factors in decision making. These criteria serve as a general guide and are important to be a rigid list. Not all criteria must be fulfilled to establish scientific causation. In Hill's criteria, there are actually 9 guidelines for judgment whether an association is causal. The temporal relationship, the strength of association, the dose-dependent relationship, the consistency of findings, the biological possibility, the coherence of the association, the experimental evidence, the specificity of the association, and the analogy. The temporality refers to the necessity that cause of the risk factor precedes the effect of disease in time. This criterion is inarguable. Only if it's found that C, the cause cannot precede D, can we dispense that the cause or hypothesize that the cause could cause the disease. Of course, observations in which the cause precedes D will provide no evidence for or against the hypothesis in some circumstances. For example, the cock crowed, then the dawn came. It is important to pay attention to the time interval from the cause to disease in chronic disease study. Obviously, the disease cannot be attributed to asbestos if one person exposed to asbestos for 3 years developed lung cancer. Among epidemiological study design, temporal relationship is shown more clearly in the experimental and cohort study than in the case control and cross-sectional studies. In this figure, we are showing the temporal relationship between thalidomide sales volume and the incidence of focal media in the in 1950s to 60s, which suggests the causal association between thalidomide and focal media. The strength of association in epidemiological study is measured by the size of the effect, expressed by the relative risk or relative rate or odds ratio. The stronger the association, the more likely the causal relationship. For example, we can see that in the text that the smoking more than 20 cigarettes per day will increase the risk of laryngeal cancer with the relative rate as 20. However, some strong association may be entirely or partly due to unmeasured confounder or other source of modest bias. Of course, once the confounding factor is identified, the association is diminished by controlling for the factor. We can deduct this confounding factor is the cause. For example, a strong relationship between Down syndrome and birth order was observed, which is confounded by the maternal age. When the researchers acknowledged that a weak association does not rule out a causal connection. Today, some associations, such as those between smoking and cardiovascular disease, or between environmental tobacco smoke and lung cancer, are accepted by most researchers as causal, even though the associations are considered really weak. Therefore, a strong association is neither necessary or sufficient for causality, and a weak association is neither necessary nor sufficient for the absence of causality. As in previous epidemiological studies, people usually suggest that when the relative rate or odds ratio larger than 3 in the cohort or control study could provide a very strong support for the causation. The dose-dependent relationship is also very important for causal inference. As the dose of exposure increases, the risk of disease occurrence also increase or decrease. For example, more smoking means more cigarette exposure and more tissue damage, hence more opportunity for carcinogenesis. In a word, people who smoke the most cigarettes are more likely to suffer from lung cancer. In the cases of focal media, the condition varies in different regions and years according to the sales volume of thalidomide. Associations that do show a monotonic trend in disease frequency with increased level of exposure are not necessarily actually causal. Confoundings can result in a monotonic relationship between a non-causal risk factor and disease if the confounding factor itself demonstrates a biological gradient in its relationship with disease. The relationship between birth rank and Down syndrome shows strongly a biological gradient that merely reflects the progressive relation between maternal age and the occurrence of Down syndrome. A clear dose-response relationship can increase the likelihood of causal association, although the absence of the dose-response relationship does not necessarily rule out a causal relationship. Consistency refers to the repeated observation of an association in different populations under different circumstances. Causality is more likely when the association is reported by repeated observations. For example, more than 100 studies over the last 40 years demonstrated that smoking can increase the risk of lung cancer. Lack of consistency, however, does not rule out causal association, because some causes produce effect only under unusual circumstances. Blood transfusion can cause infection with human immune deficiency virus, but they do not always do so. The virus must also be present. Consistency may only be apparent when all the relevant details of the causal mechanism are understood, which is very seldom. Biological plausibility refers to the scientific likelihood of a causation. That the start point is an epidemiological association. To determine if an epidemiological association is causal, one must consider its plausibility. That plausibility refers to a consistency with what is known about a particular disease, including the biological knowledge, the prior beliefs or knowledges. The causal relationship between hyperlipidemia and coronary heart disease can be supported by pathological evidence of coronary atherosclerosis and animal experimental results. The term coherence implies that a causal association should not conflict with disease natural history and biology. Here's an example for coherence, such as the histopathological effect of smoking on bronchial epithelium, or the difference in the lung cancer incidence by sex could reasonably be considered an example of plausibility as well as coherence. The distinction appears to be a very narrow one. Absence of coherence cannot be taken as evidence against causality. The presence of conflicting information may indeed refuse a hypothesis, but one must always remember that conflicting information may be an error or a misinterpretation. An example is the inhalation anomaly in the smoking and lung cancer, which is the fact that most lung cancer among smokers seem to be concentrated at site in the upper airway of the lung. Several observers interpreted this abnormality as evidence that cigarettes was not responsible for the majority of the cases. However, other observations suggest that cigarette-borne carcinogens were deposited prefrontally where most lung cancer cases were observed, and so the anomaly was in fact consistent with the causal role for cigarettes. In some hawks, coherence is interpreted as the consistency of distribution. The determinants of distribution should be consistent with geographic and other distribution of the disease. Experimental evidence is also needed for a causal relationship. Three types of experimental proofs can be established: the experimental in human subjects using the risk factors which is set on a label, the experiment in animals which may not be relevant to human beings, and the cessation effect of the exposure. This experimental evidence refers to the experimental or semi-experimental evidence obtained from reduction of or elimination of putative harmful exposure and seeing if the frequency of disease subsequently declined. Here called this structure passive evidence of causality. It can be faulty, however, as a semi-experimental approach can be confounded or otherwise biased by a host of concomitant secular change. Moreover, even if elimination the exposure does causally reduce the frequency of disease, it might not be for the etiological reason hypothesized. The draining of a swamp near a city, for example, would predictably and causally reduce the rate of yellow fever or malaria in that city the following summer, but it would not be a mistake to call this observation the strongest possible evidence of a causal role of meiosis. Specificity is that the cause leads to a single effect, not multiple effects, and that an effect has one cause, but multiple causes. Specificity indicates that a single exposure is associated with only one disease. Specificity strengthens the evidence for causality, but the lack of specificity does not rule out causality. For example, the association between smoking and lung cancer is not specific because the causal factors of lung cancer is not merely smoking. However, we cannot deny the causality between the smoking and the lung cancer because of the existence of other evidence. Analogy is a part of this criterion testing that if the exposure to association is like other established causal associations, the case for causation is strengthened. But the absence of such analogies only reflects a lack of imagination or experience, not the falsity of the hypothesis. We might find naive here's an example in which reasoning by analogy from the Xerodomian and rubella tragedies made it more likely to him and other medicines and infections might cause other birth defects. But such reasoning is common. We suspect that most people find it more credible than that smoking might cause, say, stomach cancer because of its association. Some widely accepted as causation with cancer in other internal and gastrointestinal organs. Here we see how the analogy criterion can be at odds with either of the two specific criteria. The more apart the analogy, the less specific the effect of a cause, or the less specific the causes of an effect. We should bear in our mind that the process of causal inference is complicated, as aforementioned nine criteria. The most important one is temporal relationship. Strength, specificity, and consistency are not necessary criteria. However, they are also very important to increase the possibility for causal inference. In the end, I would like to emphasize some of the key points in today's course. The first is that we need to understand the definition of the cause of the disease. The second is that we will need to know the methods in causal inference, which is the Mills canon. And we also need to understand the 9 guidelines for judgment whether associated is causal, which is the Hill's criteria. That's all for today's lecture, and thank you for your time.